Okay, hi there. Jay Shendure from the University of Washington. Thank you for the opportunity to present here at this conference, this virtual conference, and tell you about some of our work. So I'm going to try to squeeze in two different but highly related topics. And I think hopefully by the end you'll get at least some flavor for how they're related to each other. But the broad theme here is trying to reconstruct the process through genomic methods and two very different approaches, but also two very different applications, and in particular, the first one being cancer, where I'll talk you through some recent work around trying to reconstruct the evolution and development of cancer in a orthotopic model of pancreatic adenocarcinoma. And in the second part, I'll tell you about trying to reconstruct this process, also a process of cell growth and diversification, but the process of natural biological development. Okay, but just to dive in here on this first topic, which will be reconstructing cancer. This is work that's been collaborative with Chris Legner's lab at UPenn, and in particular involving also Aaron McKenna, who was a trainee in my group, now has his own lab at Dartmouth. But really led and driven by Cayman Siminoff, who is an MD PhD student at UPenn and Chris Legner's lab, and really was the one who was the driver of this work. And so the broad context here is just about this idea of reconstruction relates to this method that Aaron developed in my group a number of years ago in collaboration with Alex Shear's lab. That we called genome editing of synthetic target arrays for lineage tracing, or Gestalt as an acronym. So to give you an idea of how this works, and in the original study, which I won't really talk about today, we were applying this to development, but the work I'll talk about today, we're applying it to cancer, but the concept is basically the same. We have engineered into a single cell. In the original study, this was a zebrafish zygote. All the reagents for CRISPR-Cas9 editing, but then also a barcode of CRISPR-Cas9 target sites, a synthetic barcode. And basically, this barcode accumulates edits. And because these edits are essentially occurring consequent to double-stranded breaks and non-homologous end joining, you can get a diversity of outcomes. And those outcomes are irreversible, and they're passed on to daughter cells, and then to the daughters of those cells, and so on and so forth, in an irreversible cumulative way. And because different mutations accumulate in different sort of sub-branches of this overall tree, as time goes on, one can at the end sequence these barcodes and try to relate cells to one another based on the barcodes based on the edits that they share at this barcode. So for example, if these were the barcodes that we sequenced, we might be able to phylogenetically reconstruct their relationships to one another on the basis of shared edits. Right, you get the idea. And so we applied this method in this original paper a couple of years ago now to zebrafish development. We're able to construct some course trees of its development and kind of showcase the power of this approach, which I think has also been picked up by a number of other groups now. Okay, but what about the challenges, right? So there are certainly challenges. So amongst those, in the context of trying to adapt Gestalt to mammalian system were these, right? So proliferation is slower in mammalian populations. So often like our barcodes would get completely edited out, which limits our ability to derive information from the patterns that we see. In the case of zebrafish, we were simply injecting guide RNAs and Cas9 into a single cell zygote that had this barcode. That's not possible with mammalian systems. We have to encode all of the components genetically. And I think really to maximize the value of this kind of approach, you want to know something about the cells in which you're reading out each barcode. So for example, a powerful approach would be to have a single cell transcriptome, which is a technology that's really blossomed in the last couple of years, and to be able to say something about the molecular states of the cells in which barcodes are observed, and then we can relate them to each other and so on and so forth. And so that's really what we were setting out to do here. And so some of these problems we solve with kind of engineering, basically, the components of the Gestalt system. So in particular, for example, how we introduce these guide RNAs that are going to be editing that barcode. And so we tried really a diversity of different approaches, but really, ultimately, we're able to succeed in developing approaches where in a compact way, we could encode lots of guide RNAs that then stochastically were editing different targets. And then, Cayman went on to also engineer other components of this system, and ultimately come up with something that we gave a slightly amended name of Max Gestalt, which stands for multiplexed, because we've got many targets here, activatable because this is now an inducible system that we can induce with doxycycline, and the resolution is such that we are able to get both clonal and subclonal information by virtue of integrating lots of these new Max Gestalt barcodes to different locations in the genome, along with kind of a random tag, which in a nutshell enables us to kind of lineally mark a bunch of cells, inject those into a mouse and allow them to develop into tumors, and then be able to trace back not only to that founding cell of origin, but also to the diversification or subclonal information that had occurred as the progression occurred in the mouse. And so the broad goal here, I think, in the context of the field is really, and I think people on this who are listening to this, I think probably appreciate this without much of a background, but is really to try and understand as a tumor develops, what are the lineal relationships between different subsets of metastatic and primary tumor cells, and how those relate to one another, both lineally as well as in terms of molecular state. Okay, so coming back, I briefly introduced this before, but to the specific kind of system we picked and why we picked it. So in particular, Cayman was really interested in this question of what drives metastases. And as I'm sure many of you know, incredible amounts of effort has been put into identifying the genetic drivers of cancer. And of course, this is met with great success, with drivers identified for nearly every major human cancer, and clearly lots still to learn there. But there hasn't been the same kind of success in terms of recurrent driver mutations in relation to metastasis specifically, all right? And we don't see that recurrence the way we do for primary tumors. And so this leads to the question of whether some of this progression really may be epigenetic, or something that would be evident at the transcriptional level, even if you didn't see sort of specific mutations kind of underlying what happens between when you go from the primary site to being a metastasis. So so really, the other kind of goal here was trying to understand in pancreatic cancer, where metastasis plays a big role, right? And picking up things early can lead to really pronounced differences in survival. So this is the specific system that Cayman got set up in collaboration with Bobby Nograd and Ben Strager, which involves this orthotopic injection of cells bearing these KRAS and P53 mutations into a mouse, and allowing those to then engraft and expand, and you get metastases at multiple sites. And we start doxycycline induction to basically trigger the Max Gestalt recording. And then after the mice reliably die, and then we can collect material from various sites, sort out the PDAC derived cells by virtue of a GFP marker, and then perform analysis of these Max Gestalt barcodes, but done in such a way that we also, with 10x sequencing, 10x genomics profiling, also get the single cell transcriptional profile of each cancer cell in addition to its barcode. Okay, so and then on the analytical side, what we're looking for is kind of, when these orthotopic injections occur, we're throwing in about 30,000 cells into the mouse. And so one question is, To what extent are the metastases that result deriving from, how big of a subset of those 30,000 are actually contributing? So that's just this what we're referring to on the left with clone discovery via these, the barcodes that kind of mark the cells. And then in addition to that, we're trying to reconstruct the evolution of the barcodes within these clones, and then finally trying to integrate this with transcriptional information that we get from the 10x data. Okay, so let's actually just look at like what happens here, right? So we've got this is now a summary of what is seen with respect to clones and their contributions to observed cells at different metastatic sites as well as at the primary site. And so the main message here, so every column here is a different clone. And just to reiterate this, I maybe didn't do a great job of explaining this, but we're taking these max Gestalt barcodes and we're introducing them lentivirally to integrate at many sites within the PDAC cells that are going to get injected. But the combination of these what we call static barcodes, the non-evolving component, or just kind of a degenerate tag, kind of similar to other clone tagging approaches, tells us, what that uniquely mark a cell, right? Some combination of those. And so what we're really seeing here is that out of those 30,000 cells that get injected, it's really a tiny minority that end up contributing to what we observe as ultimately cells that are engrafting at the primary tumor as well as at the sites of metastasis, right? And the bottleneck is significant at the primary site, and it's much greater at the metastatic site. So here, for example, we're seeing this single clone, mouse clone 1 on the left there, that's really dominating at all of the sites that we're sampling, with minority contributions from other sites, right? And this is all from one mouse on the top. We also did essentially repeated the experiment on a second mouse, and you see something similar where maybe there's a bit greater diversity, but still it's really just a handful of clones out of these 30,000 getting injected that are engrafting and dominating. Okay, so now the other observation here. So this is what we're showing here on the left. And actually, all of these images are really the same UMAP. And so UMAPs are a way of representing single cell data in a visually tractable way. So every point here, these are dense fields of cells, but every point is a single cell. And they're colored in different ways in different plots. On the left, the top left, they're colored by the clone identity. And we see integrated data from actually both mouse 1 and mouse 2. And what we're really seeing there is kind of what we saw on the last slide, a few clones, M1.1 and mouse 1 and M2.2 and mouse 2 are really dominating the field with minority contributions from lots of other clones. But what's also interesting here is that if you kind of take the amount of geography that's eaten up on this UMAP as a proxy for transcriptional variability, there's a lot of transcriptional variability within these large clones, right? Almost more perhaps than the transcriptional variability amongst the non-aggressive clones that are also detectable. Okay, so that's one interesting observation here. We can also calculate kind of an aggression score that you see on the right based on some the aspects of how quickly these are inferred to be proliferating. And there again, you see kind of you can see the dominant clones, but you can also see that within those dominant clones, there's a lot of transcriptional heterogeneity. So, okay, and so kind of the other question we can ask here is, as I mentioned earlier, we have the 10x transcriptomes of these cells, so we can look at molecular features that are associated with clones that appear to be really successful in terms of metastasis, and clones that maybe are detectable in the primary tumor, but not necessarily in the metastases, and that's kind of the clones that would occupy the circled region there. And as you might expect, in that circled region, the quote unsuccessful clones that are still detectable, we see markers of epithelial genes. And in contrast, if we look at the region, so this is now looking just at mouse 1, you can see kind of the big M1.1 clone there. What the genes that are expressed out there are really the mesenchymal genes. And this is kind of consistent with some sort of epithelial to mesenchymal transition being related to this transition to metastatic success. But obviously this is not a binary process. This is a continuous process, and we can in fact see kind of signs of that continuity in the data, right? So here we've put the cells through the monocle package, which is developed by my colleague Cole Trapnell, and assigned every cell a pseudo EMT score, which is analogous to a pseudo time score, except the scale here is increasing transition to being mesenchymal and instead of time. And so you can see this progression from the most epithelial on the right to the most mesenchymal on the top left in relation to this UMAP. Okay, and then we can, in relation to that, we can then plot the expression of different genes along that pseudo EMT trajectory and come up with a continuous view of the EMT process, right? As things progressed from epithelial to mesenchymal in the context of this, development of a tumor in this one mouse. Okay, and so here's kind of another view of that where we really condensed things across thousands of differentially expressed genes across the pseudo EMT trajectory and grouped them by different clusters or patterns, which E and then H1 through H4, so hybrid 1 through hybrid 4, so early hybrid to late hybrid states. So going top to bottom epithelial, then H1, 2, 3, 4, and then finally the most mesenchymal state, right? And then different pathways, including different signaling pathways, but also, OXPHOS and different glycolysis, et cetera. You can see those pathways active at different moments along the time course, right? Which is really interesting. And so this is, again, kind of coming back to this point that, we're viewing EMT as kind of this more continuous process, and we're able to dissect it by virtue of kind of having all these states represented in some form within this one mouse. Okay, and then, I think the naive assumption, reasonable assumption, would be that kind of the advantage for metastatic success would linearly increase or somehow increase, monotonically increase across this pseudo EMT trajectory. And we wanted to actually investigate that and see whether that held up. And so as part of that, we turned to the actual lineage data, where we're no longer just looking at kind of the, which clones are achieving success, but actually looking in a little more detail at which, within within any given clone, how the barcodes, the max gestalt barcodes are evolving, right? So this is kind of more of a high-level summary of the number of barcodes that we pick up, how often we're able to detect barcodes and to find edits. And we actually do see quite a lot of editing with the DOX induction of the CRISPR target sites across the 28,000 single-cell transcriptomes that we sampled. And then we can use those, that barcode information to reconstruct trees, phylogenetic trees. And so here at the bottom, we're showing clade M1.1.310. And so this is a subclade of clone 1 in mouse 1, that dominant clone that met with metastatic success. And you can see that we have greater granularity with respect to trying to build a tree from something like this. And then we can look at, we have the ability then to kind of look at the tree in more, subparts of the tree in more detail, right? And so one way of visualizing this kind of data is those trees, but that's really not compact enough to really get a full view. So Cayman worked to develop this other approach, which we really like to kind of see the whole picture. And so in this kind of circle diagram, every, these big, bold outline circles represent the clones, right? So the different cells that were injected. So M1.1 is that big circle. And then the more lightly outlined cells represent kind of subclones or sub-subclones or sub-sub-subclones within that and within that major clone. And here they're colored by the site at which they were found. Each cell was found, right? And so you can see, this M1.1, as we talked about earlier, is found at a diversity of different harvesting sites of metastasis as well as in the primary. Whereas if you look to kind of the smaller clones, you can see that aqua color more frequently, which corresponds to the primary tumor. So those are the, quote, less successful clones in terms of getting out to the mets. Okay, and again, this is all kind of part of a different way of representing the same information that we see on the UMAP. So now let's kind of take clone 1.1 on its own and look in a little more detail. And so one of the interesting things that we're able to do is to by just looking at M1.1 on its own, we can for these different subclones and sub-subclones and so on and so forth, we can calculate kind of a dissemination score, right? It's based on the success of that clone or subclone or sub-subclone in getting out to different sites, right, and metastasizing. And what you can see here is that these smaller ones that are kind of labeled as root clades, right, correspond to things that generally have very little metastatic success, right, and are much smaller and tend to be in the primary, right. And what's interesting is we here the bottom and the top are essentially the same, just colored differently. The top is colored by this dissemination score. The bottom is colored by the pseudo EMT score. And in general, you can see a correlation, right? You see that the smaller clones tend to have earlier pseudo EMT scores, tend to be more epithelial, and the larger clones tend to be more mesenchymal. But it's actually interesting to see that like the largest clones are not dark red on the bottom, right? They're not the most mesenchymal of all the cells sampled. In fact, the most mesenchymal cells are not as successful, right? So that was pretty interesting. And so we can look at this a different way, just to kind of summarize that information on the top right. We've got the pseudo EMT score going from left to right. And in the y-axis, we've plotted the dissemination score of different subclones and sub-subclones. And what you see clearly there is what you kind of can also see on the left in a more anecdotal way, is that the dissemination, the success of clones peaks for those with intermediate EMT scores, right, pseudo EMT scores. And in particular, this kind of late hybrid score is the one that tends to be the kind of what would appear to be the winners. And the root clades tend to have these, earlier scores as you would expect, right? And then the ones with the very high EMT scores are less successful than those with intermediate, in particular, late hybrid scores, right? And so again, going back here, this conflicts with our original intuition in the sense that this H4 state, this late hybrid state, appears to be the one that's the most successful. Okay, and then another, just to, again, this was all from a single mouse, right? But we can take these scores and we can basically use the pseudo EMT that's kind of defined on these data to then score individual basically RNA-seq data from the TCGA project and pancreatic ductal adenocarcinomas, and basically try and decipher, or assign a pseudo EMT score effectively to individuals. And what we find is that the hazard ratio, right, for mortality actually peaks for human tumors that are metastases that are associated with this kind of late hybrid score, right? More so than the ones that are fully mesenchymal. Okay, so that's kind of the main arc of the story here, and this is not kind of the first description of this hybrid EMT state, right? This has been looked at before, and here's kind of some characterization of this from the literature. But here, I think one thing that I think is really nice about the study is that we do get this continuous view and a whole transcriptome view that allows us to look at what's going on transcriptionally. What we see is that it's not simply a balance between epithelial and mesenchymal that tips in one way or another. Rather, you see both programs, so metabolic programs and signaling programs and transcription factors that are kind of unique to these intermediate states, right? And so that's interesting. Okay, so that's kind of the high level story here. Of note, we have done this in a second mouse that I alluded to at the beginning, and we do see a similar picture here, right, where we again have a dominant clone, this M2.2 in this case, that has this late hybrid state, and once again kind of the highest hazard ratio with in association with it, right? One interesting kind of just anecdote I'll throw out there about this second mouse that we're still in the process of investigating. So on the left is the mouse 2 kind of evolutionary tree, so to speak, and you can see it's complex, but you can see all the diversity there. But something that was interesting is some of the top ranked genes associated with subclonal dissemination were these S100 A genes, this large family of genes that as a potentially an autocrine or a paracrine signaling molecule. And what was interesting was that it wasn't like that dominant, that this was the association of these genes with dissemination was restricted to a single subclone, or a single clone, sorry. Rather, we saw many clones, right? And these are kind of independent cells in the original injection. And yet many of them, our expression of these S100A genes is associated with subclonal dissemination. And so what this suggests to us is that there is crosstalk between these independent clones, and they appear to somehow be kind of fostering one another's success, or at least the subset that are expressing these, which would be consistent with effectively paracrine signaling or something like that. Okay, so high-level conclusions for this first part, right? So in this metastasis competent model, the vast majority of clones that were injected orthotopically were not successful metastatically, and only a few clones were dominant. Within dominant clones, even within dominant clones, it was rare subclones that were dominant, which in turn points to this idea that there's continuous evolution, strong selective pressure going on, especially given how that these are pretty fast experiments. Subclones exist along this exquisite EMT continuum that we can pull out of this kind of data, and this is a continuum that is not monotonic in its relationship to aggression, and rather we see peaking of aggression at these late hybrid states that are associated with unique metabolic and transcriptional programs that do not comport with a simple balance of epithelial versus mesenchymal. And so somehow these states are emerging very quickly from natural selection from this large pool of epithelial ancestors that are getting injected, right? And then once again, tying this back to the human data, we have this late hybrid state does predict worse survival in human pancreatic cancer. So again, really fun story, and a pleasure to be involved with this work that again was really driven by Cayman Simonov, really remarkable young scientist. And if you want more details, just came out in Cancer Cell pretty recently. Okay, so now I'm going to turn to the second part of the talk, and going back here, again, just quick acknowledgments. Going back here, as I mentioned, this second part relates to development, in particular development in the mouse. We're going to be switching from talking about lineage tracing in the sense that I was talking about it in that first part, and rather relying on some different approaches for reconstructing development, in particular, more along the lines of these pseudo trajectory approaches that Cole and colleagues have developed. Okay, so now, so again, shifting gears here and focusing on reconstructing development. So the broad inspiration here is John Sulston, and this is a lineage tree, right? Much like the trees we were trying to reconstruct in the last part, but here it's a reproducible tree that occurs the same way every time and gives rise to the remarkable little nematode C. elegans, always happening the same time and deciphered by John and colleagues through painstaking observation under a microscope over the course of several years. And so, 30 odd years have passed since this, but we still don't have equivalent maps, lineage maps, or really, a comprehensive understanding of the relationship of cell types to one another in more complex model organisms such as the mouse. And, the reasons for this are pretty obvious. Mice are not transparent. Their development is variable, not invariant, and it's a question of scale. We're now talking about billions of cells rather than 1,000. Nonetheless, we can try and hopefully bring to bear technologies that have emerged, only in the last decade or so. And so I think in the long view, this is what we would like. We would like to get a global view of mammalian development, inclusive not only of lineage, but also of the molecular states of cells, knowing where they are spatially through time, and inclusive not only of states with respect to gene expression, but also chromatin regulation, signaling pathways, so on and so forth. So really, kind of the view we have of worm right now, but for something like a mouse or a macaque or a more complex organism, as a better model for humans. So for this, to the question of scale and the massively different scale between worms and mice, we're really leveraging and having contributed to these developments and technologies over the last couple of years in the space of single cell genomics, right? And really, this is just a 10-year time scale or less than that, where the numbers of cells per study has been growing exponentially, right? And it's not just one technology. There's a diversity of technologies kind of reminiscent of the early days of NGS. Our lab has, in particular, in collaboration with Cole Trapnell and Frank Steemers, previously at Illumina, has invested in this paradigm of single cell combinatorial indexing. And this is an approach first developed by a former postdoc in my group, Darren Kusanovich, now at University of Arizona, and Riza Daza, research scientist in my group. And it's a pretty simple concept, and it's now 5-6 years old, and so I'm not going to go through this in exquisite detail for reasons of time. But the basic idea is that one is pooling and splitting and pooling sets of cells or nuclei, and performing molecular barcoding in a well-specific way at each of these rounds. And the take-home consequence of this kind of a workflow is that without ever actually isolating any single cell in any well or droplet or compartment, we are able to label its nucleic acids with a unique combination of barcodes, such that when we sequence those barcodes together with a transcript or a portion of its genome, we can group the reads that derive from the same single cell, and we can do this reliably. And the advantage of this pool split approach over other approaches is that in principle, it scales exponentially. So for a linear increase in the amount of work that's done, one can in principle tag and then profile exponentially growing numbers of cells. And I think this is, and the other big advantage here is that, it's pretty straightforward molecular biology and just wells and plates and pooling and splitting. And so it lends itself well to the adaptation of a variety of, now conventional genomic methods to single cell profiling. And so this is just a list of the various methods that we, former trainees or others in the field have now succeeded in adapting this combinatorial indexing paradigm to enable exponentially scalable single cell profiling. So, so we in our lab in particular, again, in collaboration with Cole, Eileen Furlong, Bob Watterson, others, have really been trying to take these tools and apply them to systems of embryonic development in a variety of model organisms. And so I'm in particular today going to focus on what remaining time I have on the mouse. And we didn't start with the mouse, we started with the worm in collaboration with Bob, where this first version of Sky RNA-seq, called Sky RNA-seq 2, because we have 2 rounds of this splitting and pooling, was developed by Junyu Kao, shown there, who was a graduate student with me, is now an assistant professor at Rockefeller University. And he was able to perform the first kind of whole organism shotgun single cell profiling, in this case of C. elegans, for which 40,000 cells were enough to get kind of 50x cellular coverage, kind of the equivalent of genomic coverage, but we're effectively covering every cell in the organism, an average of around 50 times profiling. And Bob and others have gone on to do much more extensive profiling of worm development. But Jun, teaming up with Malte Spielmann, a postdoc in the group who's now got his lab in Berlin at the Max Planck, turned their attention to the mouse. And also, Jun developed a 3-level version of this Sky RNA-seq protocol. And it's really when you get to 3 levels that the exponential aspect of the paradigm really starts to shine. And so with 3 levels, 384 wells in each of these rounds of indexing, one can theoretically encode as many as 50 million different combinations of barcodes. And so Jun was able to profile 2 million cells in a single experiment that he sat down and did kind of himself over the course of a week or so. And working with Malte, as I mentioned, they really focused their attention on mouse development. And in particular, it's an incredible process, right? As we all know, over the course of a couple of weeks, you give rise to this functioning organism with all of these organs and systems. But in the space of all of this that's happening, Malte, who was a mouse developmental geneticist, really helped us narrow in on this 4-day window between E9.5 and E12.5 when a whole lot happens, right? In particular with the mouse, kind of exploding in size and complexity. And so this is what we applied those 2 million cells to, profiling to. And we're able to put every embryo goes in a different well at the beginning of the experiment. And so we actually know, not only do we get single cell profiles, we're able to know what cell goes with what embryo from the starting point. Okay, so any data site like this, the annotation is an art form, and lots of credit to Malte, who worked through this very painstakingly. This was our initial effort to annotate the data. So every point on this graph is one of 2 million cells. And this is a TSNE, kind of the predecessor of UMAPs in terms of how you visualize this information. And we see all the things we expected to see at about the right proportions. So that was very encouraging. But we can also delve into more detail and look for very rare cell types, part of the motivation for going so deep. So for example, the apical ectodermal ridge is a cell type that drives limb development. It's supposed to be transient, appear and then disappear. And so we can look to see if we can find it. And so, out of this, this massive UMAP, there's lots of clusters. One of these clusters corresponds to all epithelial cells. We can pull those out and cluster them on their own, and Malte them would go through and try to annotate these and can do a reasonable job of it. And indeed, we see a cluster that looks like it is the AER, this transient, very rare cell type. And we know this because it's expressing FGF8, which is a very specific marker of it. And it's also transient. It appears and then disappears in the time course that we were looking at here. It's only 1,000 cells that we see out of 2 million, but we can find it here. And I think this kind of illustrates the value of going very deep, especially for a complex process like development. Okay, and then if we can also leverage this kind of data to then say, okay, well, we know FGF8 is a great marker of the AER. Can we find other great markers of the AER? And we look for other genes that are specific to epithelia of the AER in this data. We can go back and check them, and indeed they appear to specifically mark the surface of the developing limb bud at the right time point in development. Okay, so one of the challenges here has been that this is an enormously complex and unwieldy data set to try and analyze. And so the next couple of slides, I'm just going to try to illustrate how the choice of algorithm that you use and your approach to it can really influence how you see the data, right? So this is showing one of the limitations of our data as we originally analyzed it and wrote a whole paper around, which is that when we get these clusters that make a lot of sense, we still see scattering of developmentally related cell types. So all kinds of things that we know belong in the CNS are kind of scattered across this diagram. So concurrent with us trying to work through the data, Cole's group and Xiaoji Q, shown there, developed this updated version of his monocle package called Monocle 3 that did a lot of different things. But one of those things was to take advantage of this new UMAP algorithm from McInnes and Healy. And so in this next slide, we're looking at the same 2 million cells, but I'm just going to play a video that Cole made of transforming that data set. So the same 2 million cells, but just moving where all the cells are into 3 dimensions and now analyzed with Monocle 3. And the take-home messages here is that you get a very different view of exactly the same data. One, 3D never hurts. It gives you, it's fun to kind of zoom around and look at it this way. But also, as I'll show in a second here, the organization of the data is much more gratifying, right? We actually see that the groupings of the cells are more kind of along the lines of what we would expect. So all of, a little cluster for the lens, all of hematopoiesis in one place, different subsets of neural crest, endothelium, epithelium, hepatocytes are floating there above the entire mesenchyme, and then all of the neural tube in one place. And so that was a much nicer way to look at the data. And if we drill in further, we can see, even greater, more granular levels of complexity that are better visualized this way. So here, just taking epithelial cells and reanalyzing them with Monocle 3, we see there's a lot of richness there, even that one cluster, where you've got, lots of different subsets of epithelia, as you might expect. But moreover, even though the algorithm does not have access to the underlying timestamps on the cells, we see this very nice progression from 9 to 10 to 11 to 12 to 13 in terms of the way that the data is kind of self-organized as kind of little layers of almost like a triple-decker sandwich. And so the pockets of orange here correspond to progenitors that were presumably present at E9.5 and then branching out in different ways in time over those 4 days. One maybe unsatisfying thing here is that we don't get a nice tree. We don't get a single unifying tree, which we know development to be. But that's partly our fault because we started at E9.5, right? And so a lot of development has already happened. A lot of these cell types are already well separated from one another. So we shouldn't necessarily expect to see that single origin that we might have if we'd started at E0. So on that point, and this work was published, in 2019. On that point, one thing that we spent a lot of the time in the years since then, and during the pandemic, has been to try and go back even further, right? And in particular, leveraging the fact that while as our work on E9.5, E13.5 came out, other groups published data sets which stretch back earlier in development. All the way back to E3.5, just coincidentally, even without any coordination. And so CXQ, who's a brilliant graduate student in the group, took this on as a rotation project and turned into a thesis project to try to connect all these data sets together and build kind of a unified map or tree of early mouse development. And so the challenges here are technical in large part, right? Because these data sets were generated with different technologies by different groups, right? And that can make it very hard to piece it together. And so CX tried a lot of different things and came up with a reasonable approach that I'll summarize here. So we really just take every time point and study it on its own to define cell types and to annotate those. And then we take every pair of adjacent time points and try and relate them to one another. So if we take a pair of adjacent time points, we can actually correct for batch effects and technological artifacts and try to then co-project them to a single embedding space. And there are increasingly sophisticated tools for this, like Harmony and Seurat. And when we do this, we get an image where the data often kind of go together. And then we can try to make inferences based on proximity in high dimensional space for when new cell types appear, trying to figure out who their ancestors were. We call them pseudo ancestors because it's not explicit lineage tracing, but in many cases, we do see kind of a nearest neighbor that makes sense. And so, we can do this systematically across every pair of time points. And just to kind of walk through an example in a little more detail, here we have E5.5 epiblast, and we co-embed that with E6.5 data. We can see a new cell type, the primitive streak, appearing, and so we can relate that to the epiblast as its likely origin. And then when we go just another 6 hours out and co-project E6.5 with E6.75, we see the appearance of the nascent mesoderm, and we can infer that the nascent mesoderm came from primitive streak rather than epiblast. And then if we go out another 6 hours, taking E6.75 and co-embedding with E7.0, we see additional cell types and can infer their likely pseudo ancestors. And so that, we can do this over and over again and build kind of a pseudo tree. Key point here, this is, again, I mentioned this earlier, these are trajectories, not lineage, like we did with Gestalt in the first part of the talk. We're still developing the tools to really do the Sulston-like experiment in mouse, and I think we'll get there, but it'll take a little more time. But nonetheless, there's a lot we can do just with single-cell RNA-seq data. And so this is the resulting tree that comes from that process, and applying it to every pair of these 19 consecutive time points, each of which is kind of a column here. And then the edges here, and this is effectively an acyclic directed graph, corresponds to edges where we do infer some kind of putative relationship or potential relationship. And the things I like about this approach are it's data driven, it's automated, and yet we still are able to get a reasonable view of a development that largely, not perfectly, but largely corresponds to what we might have expected, including kind of organization of the data, self-organization into germ layers. We're able to then, the germ layers are shown kind of on the colors on the right, and we're able to kind of infer back, paths that might have led to different cell types. So I'm realizing probably I packed a little too much into this presentation. Oh, so sorry, just one example here of in general, things are organized by cell types, except when they're not, right? And this is an example of osteoblast where we appear to infer, kind of a mesodermal origin, as you might expect, but also contributions from the neural crest, which was, has been documented in the literature in the last decade or so. So I think for reasons of time, I'm probably going to go a little fast through these large parts and maybe skip some slides, but I want to kind of at least give an example of how these data are, potentially useful, or this kind of a map or organization of the information is useful. And so here, one of the things that we wanted to ask is, can we from this kind of approach go into each of these branch points and infer what transcription factors play key roles in regulating the appearance of a cell type when it first appears on this map. And so zooming in there, right, so are there TFs, for example, that you see modestly expressed in a pseudo ancestor, sharply upregulated in a new cell type, but not necessarily upregulated in any sister branches, so to speak. And looking at cardiomyocytes is kind of an example of where this, clearly can succeed. We see, basically if we apply this automated approach, you'll look for that motif of different TFs that obey those rules. The TFs that we come up with are in fact the TFs that, are known to have major roles in cardiomyocyte differentiation, like GATA5 and KX2.5 and so on and so forth. And so zooming out, like we can build these kind of, we call it like a subway map representation of the key TFs that play some role, or we infer to play a role in specific processes. And so that, the circled region here is just the cardiomyocytes, but there's lots of hypotheses that could potentially be tested, and we're in the process of kind of working towards that. So again, kind of in the interest of time and my failure to kind of appropriately calibrate to, I think, what I could fit in, I'm going to take a pause there and maybe I'll just come back briefly to mention, I'm just fast forwarding through a few slides here, but I want to mention a couple of other things. So one is that we have made a concerted effort to integrate these mouse data and these mouse trees with human development. And in particular, this is work that's all published, so hopefully this is largely intended as kind of pointers to it if people want to hear more, but working with the UW Birth Defects Research Lab here in Seattle, we were able to obtain fetal tissues from the equivalent of a slightly later window in development than we've been studying in the mouse. And applying these SKY technologies developed by Jun and Andrew and Riza and others, Darren and Riza and others, we've been able to profile really vast numbers of cells from a diversity of fetal human organs for both gene expression. These are kind of mini atlases shown here, as well as roughly 4 million cells worth of data available in this data set. And then also chromatin accessibility shown here for the same 15 tissues and roughly 800,000 cells. And kind of one of the more gratifying, please feel free to take a look at the papers if this is of interest to delve into more detail. And I'm sorry I don't have time to talk about it more today, but just kind of one analysis I'll highlight that kind of goes back to the mouse part is that we can actually integrate the human data and the mouse data quite seamlessly. And here showing these, every point here is either a human or a mouse cell. The human cells are in yellow. And as I mentioned, the equivalent human time point is a little bit later than E13.5. And indeed, we can kind of see the mouse cell types giving rise to a diversity of human cell types if you kind of follow the pseudo trajectories, so to speak. So, okay, and I think I need to stop there for reasons of time, but maybe one more slide here where I just want to make the point that I made a bit earlier, but that we're, what we're aiming for here is really this global view. And I think with technologies like Gestalt and the scalable single cell profiling, I do think we're within a few years of really being able to pull together kind of an integrated view of mammalian development, which I think will have implications for cancer as well, cells of origin and such. And really, this is something that we've only been able to do in the mouse in the worm even in the last couple of years, where even the Sulston lineage, which had lineage and spatial information, didn't really have the molecular information. It's only in the last year or two that work from Bob Waterston and John Murray and others have really pulled this together. And so this German word Gesamtkunstwerk kind of summarizes what I think we as a field are trying to do for major model organisms, which I think will support a lot of discovery going forward. And thanks, and this is kind of work acknowledgements for the second part of the talk, but I'll stop there and hope we have time for a few questions. Thanks very much.